Ok.
Ok, então, bom dia a todos que estão aqui com a gente, mais uma sessão do SIG Farmacologia e Terapêutica. Eu sou a Cristina Velar e eu estou aqui apenas para um minutinho antes de passar a palavra para o professor Paulo Guedini, que vai comandar esta sessão junto com a professora Alexandra e seu convidado, mas eu gostaria só de usar aqui um minuto para fazer uma pequena homenagem, né, um agradecimento do grupo SIG Farmaco a uma técnica muito importante, né, que é a Tatiana Patrícia Cruz, que tem sido técnica do SIG técnico operacional aqui na Unifesp, que é da onde a gente tem a coordenação aqui junto com o grupo da UFAL, deste SIG Farmaco. E a razão de eu trazer aqui para vocês é porque a Tatiana ela tem sido uma peça fundamental, né, nesse SIG. Então, eu tenho assim um agradecimento muito profundo a ela, porque logo no início, quando eu soube, né, dessa rede que se tem aqui dentro da Unifesp, com tantos SIGs que são oriundos aqui dos grupos clínicos e básicos aqui da Unifesp, a pessoa que me indicou como um caminho para começar a conhecer, né, como montar um SIG, me indicou a Tatiana, né? Então, ela foi a responsável por me dar todas as guias e depois eu, com os professores que fazem parte da coordenação deste SIG, professor Emiliano, professor Paulo, professor François e a professora Roseli, nós temos hoje, né, mais de 2 anos de sessões e ela continuou, né, fazendo parte dessas sessões, vocês devem lembrar, toda vez olhando aqui o nome dela, ela trilhou com a gente quando a gente ainda tinha, logo no início, as sessões nas salas físicas, né? Já era nossa intenção começar a ter as sessões com maior abrangência para poder cobrir mais o Brasil e ter as sessões de uma forma usando o espaço virtual. E lembro, né? Então, quem está aqui lembra, né, das nossas tentativas, e aqui na sala da Unifesp, eu, a Rose, com a Tatiana, logo no início, nos percalços, né, que para 2 anos para cá, como mudou, né, então toda essa pandemia acabou nos trazendo toda essa maior infraestrutura que faz com que o SIG tenha expandido tanto no Brasil inteiro, né, e agora até com uma extensão internacional. Então, eu acho que a gente só tem a agradecer toda a ajuda, a participação da Tatiana. Ela foi deslocada, né, para um outro hospital, onde ela vai continuar os trabalhos dela, então ela não estará mais aqui com a gente pelo grupo Unifesp. Então, por isso que eu queria deixar aqui, em nome de todos aqui do grupo SIG, Farmaco, o nosso agradecimento à Tatiana, que ela tenha um grande sucesso no próximo lugar que ela está, então o grupo que está recebendo ela, com certeza, vai ter uma excelente funcionária, uma excelente parceira, né? E falar que, então, a partir de agora, nós temos o Antônio, que está aqui, que já estava com a gente em várias sessões, né? Então, Antônio, também agradecer a você e aí repassar, né, para a Tatiana e para todos do grupo, que realmente a gente tem que reconhecer quando as pessoas elas fazem o trabalho com dedicação e muito amor, porque era isso que a Tatiana era, aqui com a gente, né, sempre muito dedicada. Então, expressar nosso agradecimento e boa sorte, né? E com certeza ela colocou para mim no áudio, continuaremos em contato e com certeza em algum momento. Então, muito obrigada. Obrigada, Paulo, pelo espaço e deixo com você, então, para continuar a sessão. Você está sem o seu fone. Ok, vou sair. Obrigada,
Obrigado,
Paulo.
Cris. A Tatiana sempre foi o nosso braço direito, né? Uma bela homenagem. Bom dia a todos. Vamos iniciar a sessão de março do SIG Farmaco, que é o Grupo Especial de Interesse em Farmacologia e Terapêutica. Desde 2018, né, nossos encontros são mensais, e esta é uma iniciativa para o uso da estrutura de vídeo e webconferências da Rede Universitária de Telemedicina, a Rede RUT, que visa reduzir as distâncias geográficas e também integrar ações em ensino e pesquisa em farmacologia e terapêutica no Brasil. Nosso grupo, né, hoje abrange todo o território nacional. Nós temos em torno de mais de 20 instituições que têm a sua estrutura física e de web conference para essas sessões do SIG, né? A mais recente, que se filiou ao nosso SIG, é da Universidade Federal do Sergipe, do Hospital Universitário de Lagarto, mas também, né, depois que essas sessões passaram a ter a possibilidade de serem assistidas e serem acessadas virtualmente pelos computadores individuais, né, ou outros meios eletrônicos, nós temos instituições representadas de todo o Brasil, que hoje abrange mais de 55 instituições participantes dos nossos encontros. Durante a sessão, solicita-se a todos os participantes que mantenham seus microfones no modo mudo, tá? Enquanto não estiverem falando. Essa medida é necessária para evitar ruídos e a perda de qualidade do áudio durante a conferência. Solicitamos que vocês deixem suas perguntas e comentários no chat, né? Que posterior à apresentação do professor Vance, essas perguntas serão levadas pela moderadora, a professora Alexandra, para discussão. Também é importante que vocês registrem as suas presenças, né, acessando o site www.rut.rnp.br/presença, sem a cedilha, inserindo o seu CPF, seu e-mail e registrando a presença através da seleção do SIG Farmacologia e Terapêutica. Também utilizando a sessão de
Paulo,
hoje...
não estamos vendo o slide. Os slides, você pode colocar?
Estou passando os slides, pois para mim está aparecendo na tela.
Para mim está aparecendo também.
Para os demais
Para mim não está. Eu até achei que você tinha decidido fazer sem slide.
Não. Nossa. Está passando os slides.
O pessoal não está vendo também.
Ué, o que está acontecendo, então?
Tudo bem, acho que continua agora nessa introdução e mais na
mas de qualquer
apresentação.
forma, a apresentação do Vance está junto, Rose. Então, nós temos que resolver isso.
Então, eu acho que você carrega de novo.
Sai carrega de novo.
Enquanto isso, eu vou continuando aqui, né? Enquanto a professora Alexandra carrega a apresentação, né? Eu estava falando para vocês registrarem as suas presenças, né? No link www.rut.rnp.br/presença, selecionando o SIG Farmacologia e Terapêutica e acessando, colocando a senha da sessão de hoje, que é 55973. O registro das presenças é muito importante, né, para a gente demonstrar a visibilidade e a importância do nosso SIG Farmacologia e Terapêutica, que tem trazido sempre discussões sobre temas importantes relacionados ao ensino, pesquisa e políticas na área de pesquisa, de pesquisa, na área de pesquisas.
Paulo, deixa eu só falar a todos do grupo. No canto direito da tela, existe um botão, foi alertado por uma colega aí do grupo, existe um botão restaurar apresentação. Eu cliquei e aí passou a apresentar os slides. Então, todos, por favor, no canto direito de baixo, no canto de baixo, tem um quadradinho, está escrito restaurar apresentação.
Eu acho que é isso mesmo, nós já começaram a comentar aqui.
Alexandra, passa o slide seguinte para ver se o pessoal está...
Eu estou no slide do registro da presença.
Isso.
Muito provavelmente eles estavam vendo apenas as câmeras e não estavam vendo a área da apresentação. Por isso que alertaram por aquele botão, é isso que aconteceu.
Ok. Vocês estão vendo o slide da estrutura da sessão?
Sim.
Sim, tá. Então agora está ok. Bom, a estrutura da sessão de hoje, né, ela é dividida em 5 momentos. O primeiro momento de boas-vindas, as informações gerais. Posteriormente, a apresentação do tema e palestrante, feita pela moderadora dessa sessão, professora Alexandra, e posteriormente a apresentação que será feita pelo professor Vance. Posterior à apresentação dele, nós teremos um período para debater, para discutir o tema, e ao final nós teremos o encerramento e o registro da participação. Seguinte, hoje a moderadora é a professora Alexandra, da Universidade Federal do Paraná, e o apresentador será o Dr. Vance, da Universidade de Ottawa, Canadá. Será a nossa primeira apresentação em inglês, né? Passo, então, a palavra para a professora Alexandra para que ela dê sequência à nossa sessão.
Obrigada, então, professor Paulo, pelo início dessa sessão. E vou fazer, então, uma breve apresentação do professor Vance. Então, o professor Vance Trudeau tem mestrado pela McGill University, no Canadá, doutorado pela University of Alberta, também no Canadá, e ele trabalha atualmente na Universidade de Ottawa, sendo que ele iniciou nessa universidade em 1998, onde ele lidera um grupo de pesquisa no Departamento de Biologia. O professor Vance tem mais de 300 artigos científicos publicados, já ministrou mais de 100 palestras no mundo inteiro sobre controle hormonal da reprodução e desenvolvimento e como isso pode ser interrompido por poluentes em peixes e sapos. O professor Trudeau é diretor também do programa de pós-graduação em biologia e responsável pela disciplina de pesquisa em neuroendocrinologia na universidade onde ele trabalha. E também foi cofundador da Sociedade Norte-Americana de Endocrinologia Comparada, já recebeu o prêmio Outstanding Achievement Award em 2017, pela contribuição significativa para o campo da ciência ecotoxicológica no Canadá, além de outros prêmios. E os seus principais interesses na pesquisa são a neuroendocrinologia, e ainda disruptores endócrinos, onde ele estuda os efeitos de produtos farmacêuticos, pesticidas no ambiente, especialmente em peixes e sapos, e tem um trabalho bem relevante sobre a fluoxetina, que é o princípio ativo então, do PROZAC, que será o tema da palestra de hoje. So, thank you, Vance, for accepting this invitation. It's an honor to us to hear you. So, please feel free to start your presentation.
Okay, well, thank you very much for the kind introduction. I'm going to tell you a part of a story about pharmaceuticals in the environment. And that's how I started this, looking at pollution, because when we take pharmaceuticals to help us with a sickness, they end up in the water. And they end up sometimes degrading and sometimes not. So this is the story of PROZAC that many of you will know, and maybe you have been administering to patients as well. So here's the title, and this is work that was going on for about 10 years, and we published an important study in 2018. So I'm going to tell you about that and perhaps some new things. And in the discussion part of the presentation, I could share some interesting data with you if people are interested. So the title, PROZAC is a transgenerational neuroendocrine disruptor of behavior and stress. And we use fish models because I like fish, but also they represent a very interesting way to look at how pharmaceuticals can affect vertebrates. And I'll show you that in a second. And I pose the question, is it time to raise the warning flag for human health? I'll give you the punchline now. A single treatment of PROZAC can disrupt fish behavior for several generations and can affect stress for even longer. So this is a work of a lot of people, and I'm going to mention a couple of people, and that's not because the other people are less important. It's just a matter of time. But many interesting and good scientists and good students from the past have worked on this project. Okay, many of you will know this compound, fluoxetine. It's an active ingredient in PROZAC. And this type of chemical, the selective serotonin reuptake inhibitors, came onto the market in the late 80s. And here you have this fluorinated, interesting compound here. So this is the first member of its family of the SSRIs. And of course, a group like this, pharmacologists will know so much more about it. But there's other chemicals now that have similar activities or may even act upon the norepinephrine system to help people with depression and other problems. This was highly prescribed because it was more quite effective for certain types of depression. And now it's quite a few, 3 decades more, almost 4 decades later, and it's still in use. So it's an SSRI, selective serotonin reuptake inhibitor. So this is where we started to get interested because serotonin is so important for many, many regulatory processes. We prescribe it or take it to change mood and social interactions. It's also very well known to regulate growth hormone and reproductive hormones in fish as well. And serotonin is also important for appetite control, especially for carbohydrate control, and even regulating what we consume. Serotonin circuits from the amygdala can impinge upon the hypothalamus to regulate CRF neurons and the stress response. So very important for all of these processes. And then when we started to think about the current studies, we came across various publications showing that serotonin is really important in the first days of life for programming of the stress axis. And this is where we started to think about the possibility that serotonin and PROZAC in particular might have long-term effects. And this was the insight of Dr. Marilyn Vera Chang, who was a PhD student at that time. And it was her insight that really got us into this long-term study. So what's the problem? For many years before, we were interested in the fact that fluoxetine is taken by patients and can end up in rivers and streams and the ocean and what have you. And in some places, this is quite an exaggerated number, but in some places, fluoxetine was reaching almost a microgram per liter. We now know that if you add up all of the different SSRIs in a medium-sized city or a large city, you will find very, very high levels of PROZAC and venlafaxine and many other antidepressants and their metabolites in rivers and streams, far exceeding a microgram per liter in total content. So it's an environmental problem, and we were studying the effects on adult fish reproduction for many years. Then came along Marilyn, and she found some studies showing that fluoxetine can be found in and around a fetus of the human. Look at the levels that are recorded. These are in women who have been carefully prescribed fluoxetine during prepartum depression. Amniotic fluid levels, cord blood levels from several different studies are all in the microgram per liter range. These are the therapeutic levels that the mother is taking and the fetus can be exposed. So this presents an issue. Serotonin is important for development, and the fetus might be exposed. Now, here's one study from a group of studies from the group of Oberlander at the University of British Columbia in Vancouver. And And this really got us interested. This one study where he was looking, and many follow-up studies, looking at cortisol in babies whose mom was taking fluoxetine or other antidepressants. And this is the salivary cortisol as an indicator of the stress status. Here is the saliva levels from a 3-month-old child, which mother is not taking SSRIs. And here is a corrected level for mothers whose babies have been exposed in utero and they have lower cortisol. So this is a very telling study. And Oberlander's group has been following these patients for many, many years. And I'll come back to some of those studies at the end of the talk when I pose the question, is it time to rethink how we are prescribing fluoxetine? Okay, so the critical question that I want to address today is, what are the long-term consequences of chemical exposure? And the example will be fluoxetine. And we're interested in it for 2 reasons. The environmental reason, this will be our low dose in the study, and from a human health perspective, that will be our high dose in the study. Does it disrupt the stress axis? And does that persist across generations? You may know that the stress response is epigenetically inherited. And I will show you that this is the case in the fish model as well. And it can persist for a very, very long time. Okay. This represents a typical teleost fish. So this is a zebrafish, which is from the largest group of fishes, the cyprinids, the carps and minnows. And for those of you that are not familiar with the fish model, the stress axis is very similar to the mammals, just a few anatomical differences, but the same hormones are involved. And I'll just take a bit of time to go through this. Here is the brain. So it's an unfolded brain compared to mammals. And the typical areas that you'll be familiar with are the green, which is the hypothalamus, and the blue, which is the preoptic area, preoptic hypothalamic region. And this releases corticotropin releasing factor, CRF, just like in humans. And if you work on rodents, the same. Controlling the pituitary gland in purple here. And the pituitary then releases the pituitary hormone called ACTH, adrenocorticotropic hormone. These are highly conserved hormones. So a stressor activates the serotonergic circuits, activating CRF release, leading to the pituitary. And a lot of this is very similar in anatomy. There's some small differences that I won't touch on today. The biggest anatomical difference is at the end here where cortisol is being produced. So in mammals, cortisol or corticosterone in rodents would be produced in the adrenal cortex. Well, fish don't have a discrete adrenal gland like mammals. The adrenal tissues, both the steroidogenic tissues, the little dots here, and the adrenomedullary tissue, so the chromaffin cells, are intermingled in this complex organ here, which we call the interrenal. This is a mixture of tissues. The kidney and the adrenal tissues are mixed together. There is no discrete adrenal cortex. There is no discrete adrenal medulla. These cells are in little pockets throughout this anterior portion of this organ. And I'll show you a picture a little bit later on. Cortisol does all of the same things in fish as it does in mammals, regulating metabolism, regulating behavior, regulating neurogenesis. So all of the typical cortisol things is very similar. It's just these anatomical differences. But this will be important a little later on. So here is the experimental design. First, day 0 is fertilization. And 2 days later, they will hatch out of the egg and they start swimming. And in the first 4 to 6 days is when the serotonergic circuits in the fish brain are developing, and when that hypothalamic pituitary adrenal axis is becoming fully functional. So this happens in about 6 days. So our exposure is from 6 hours post-fertilization to 6 days post-fertilization to cover that period of formation of the circuits, programming of those circuits, and functionality of the adrenal gland becomes very quickly set up. So you can see the advantage of a small organism like this, where the development is fairly rapid. It also means you have to work very fast to get the experiment going. So very simply, we have clean population, we breed them, and we set up the first generation. So these were fish that were in a very clean environment. Breed them. We have a control. We have a low fluoxetine. This would be something like median levels in a contaminated environment, and high levels, which would be equivalent to what's found in the cord blood of an exposed fetus. So there's a 100-fold difference here. We expose them for 6 days. Then we put them in fresh water for 6 months. And then we do different tests. A stressor test to look at the physiological response. So picking up the fish with a, I'm going to change my position here. With a net, this will increase cortisol. We did some behavioral tests, a novel tank test, an exploration of the environment, so locomotion. And we did some breeding because our background is in reproductive physiology and reproductive endocrinology. But I only have one slide about this because the embryonic exposures did nothing to breeding. This was a big surprise to us. So I will spend my time looking at the different generations, the physiological response and the inheritance of that response, and the behavioral response. So F0 is the filial generation that was exposed. F1 was the next generation, no more PROZAC. F2, no more PROZAC. F3, no PROZAC. So only the single exposure in the first 6 days of F0. Okay, this is the first piece of data that Marilyn obtained, and it was really exciting to us. So here is cortisol. So fish produce cortisol like humans. And we're looking at whole body. These are small animals. Most of the zebrafish we use would be 0.2 to 0.5 grams. So we took the whole body. We wanted to know the total status of the cortisol production. So we grab the fish and sample it. In the basal condition, so it's quickly catching as quickly and sacrificing as quickly as possible, and under a standardized stress test, which is picking it up in a net a certain number of times, etc. And you can see here, control will be always white, low fluoxetine is gray, high fluoxetine is dark on the series of slides that follow. So here you see a typical stress response, let's say around a 3-fold or a 4-fold increase after a few minutes, 30 minutes, I think. So that's great. The controls are responding to the stress. Now, in terms of basal levels, the low fluoxetine, they were exposed 6 months earlier, and they have reduced basal, and they have a reduced response. So in response to the stress, they are producing less cortisol. These are, these are males, by the way. I see that the slide changed. Males have a stronger response than females. I'll show you some female data nevertheless. Okay, now what happens? Over generations, in fresh water for the next generations. Beautiful stress response in the controls that is still reduced in the fluoxetine treatment. This is a year later, almost. Then we go to the next generation. Stress response that is reduced in those that were exposed in the F0. F3, next generation, 6 months later, you can see the basal bouncing around a little bit, but they have a stress response that in the high dose remains reduced for 3 generations. This was an incredible shock to us, and we have pursued this type of study since these data. So here's the female. A couple of things to notice from this slide. It's the same setup, cortisol and the different treatments, and in F0 to F3. So the basal levels are here. Females do have a stress response. It's a little bit less, about not quite twofold. And they're less sensitive. They have a slight decrease at the high dose. Over here, they have a stress response that's a little bit inhibited. F2, stress response, that's a little bit inhibited. F3, stress response, that's a little bit inhibited. It's still statistically significant, but is not as obvious as in the male. So most of our studies after this will be on the male. It's just a more obvious effect. So let's go back to this figure. So I've told you about the cortisol response over here, and we start looking for the site of the disruption. Is it up here, or is it at the pituitary, or is it at the steroid-producing cells itself? We did a lot of studies. We didn't find anything on CRF. We didn't study ACTH production, unfortunately, but we suspected very quickly that there's something wrong with the cells that produce cortisol. So we figured out how to inject ACTH at the right dose, and we asked the question, what is the effect of the fluoxetine on the cortisol production? So is the site of the disruption caused by fluoxetine at the adrenal cells? And the answer is yes, and here are the data. This is in males, and here's the first generation that was exposed, and 3 generations later, no more exposure. Here is cortisol again, and 3 different treatments. NI is no injection. Saline is the injection control. And we gave a small dose of ACTH, just enough to provoke a 3-fold increase in cortisol levels, so to mimic what the net stress was doing. But we circumvent the hypothalamus and the pituitary, and we're injecting the pituitary hormone. So let's go through this data. Same codes, control low and high fluoxetine. The no injection. So here is a basal level, and even at this stage, we just take the fish out of the tank and sacrifice it. You can see low levels of cortisol production in the high fluoxetine. Just injecting saline can provoke a stress response. It's a subtle stress response, but it's there. And even that stress response can be reduced by the fluoxetine treatment. Here's the ACTH response, and the high dose of fluoxetine, 6 months previous, still has an effect to reduce corticosteroid production. So we're pretty sure that the problem is something wrong with the adrenal gland. Here is 3 generations later. Very similar pattern. There's slightly different levels of the hormone, but the pattern is the same. Handling stress, so saline injection, is reduced by 3 generations previous in treatment with fluoxetine in the water. ACTH response, very nice. That is inhibited by the fluoxetine given 3 generations earlier. So here is that tissue. This is opening up the fish and looking in with all of the internal organs removed, and along the spinal cord is the interrenal tissue. This is the kidney part with some other tissues, and the head kidney, which is a mixture of the adrenal gland with the kidney. It's very small. You need a microscope to dissect it, but you break the membrane and you can pull this whole organ out. And that's what we did RNA sequencing on, to try to understand what's going on with the steroidogenesis. And here's a complicated, complicated figure, but just have a look at it. So fluoxetine modifies steroidogenesis in the generation that was exposed. And we know that cortisol is decreased. So all of these different shapes are different genes. And red is up regulated relative to control, and green is down regulated relative to control. And this is the ingenuity pathway related to steroidogenesis, so quite changed. And this is the low fluoxetine and the high fluoxetine, and the same genes are represented here. And you can see that these genes, all implicated in steroidogenesis, are altered with the pre-treatment with fluoxetine 6 months earlier. So let's grow some fish over generations and do the same experiment 3 generations later. You have a different pattern of expression, but the same genes remain different from control. So even 3 generations later, the steroidogenic pathway is altered by the treatment. So we're pretty sure that we have affected the ability of the animal to respond to stress with a single 6-day treatment. Okay, here's a simpler diagram just to pick a couple of parts of the steroidogenic pathway that you may be familiar with, and some specific genes that we found that were altered generations later. So typically, the hormone ACTH will act on its the melanocortin receptor, driving it's a G protein coupled receptor, so driving cyclic AMP and protein kinase. Many genes in the cholesterol mobilization pathway were altered. Genes involved in biosynthesis of cholesterol were altered. Then, of course, cholesterol is supposed to be imported and converted to pregnenolone in the mitochondrion, and CYP11, that very important step in steroidogenesis, was altered. Also CYP17, the hydroxylase in the ER, that will drive the production of the precursors, were altered, as were CYP17, CYP21, and even the last stage, so the 11 beta 1, the 11 beta hydroxylase was altered such that cortisol is decreased. The metabolic enzyme was also changed. So these are just cherry picking a couple of genes that were altered. So I hope I've convinced you that even 3 generations later, PROZAC can affect steroidogenesis. Okay, what does it mean? Some of you might say, well, okay, a little bit less cortisol, they're responding a little bit less, but they still have a stress response, and that is true. But I will tell you now that it disrupts their behavior, and we've proven that cortisol controls exploratory behavior. Now, we did some behavioral tests, and it's called the novel tank test. You take a fish and you put it in to a new tank, and they swim around. And you film it, and you measure speed and distance, position, time at rest, all of these different parameters that measure exploratory behavior. And we quantify that, and I will show you now that the PROZAC changes this behavior. Okay, here are males again, and this is a principal component analysis, PCA. And principal component 1 is largely driven by speed, distance. And here is the data. The high fluoxetine dose decreases the behavior over F1 till F2, normalizing by F3. This is incredible. the behavior is changed after a 6-day treatment for 2 generations. Cortisol is still changed, but they recuperate. Now, I will prove to you in a second that it's the decreased cortisol that's driving the decreased locomotory behavior. Okay, so the question, is low cortisol responsible for reduced behavior? I'm going to show you metirapone exposure and cortisol supplementation, and then wrap up for some questions. Okay, here's the cortisol synthesis pathway. Metirapone, as many of you will know, will inhibit this last stage and will decrease cortisol. And it works well in fish as well. And here is the experiment. Cortisol with the drug. So control net stress, give them metirapone for 7 days, and you have no stress response. They cannot synthesize cortisol upon the stress. So it's working. Here's what happens to their behavior. The same as we see with the PROZAC, the other chemical that reduces cortisol has the same effect, and they swim less. So chemical inhibition recapitulates the fluoxetine effect. So it's looking like cortisol is doing the job here. Here's the proof. We can rescue the behavior with cortisol. So here's cortisol again. Control high fluoxetine and put cortisol in the water. They take it up and cortisol goes up. So we've restored the cortisol and maybe even overshoot a little bit. What is the behavior? Here's the decreased behavior. Here's the rescue of the behavior. Now, we had a very nice video to show the change in behavior, but it could not be uploaded to this platform. So I'm just going to take a second and describe what these fish do. So if you put a normal fish in the tank, it swims around and explores, trying to find a hiding place or to check things out. If you give them the fluoxetine, they go down and they swim at the bottom and they stay there. They do not explore very much. It's quite remarkable. So that's what this means. They kind of go to the bottom and stay there and swim a little bit. When we put cortisol back for 3 days, they start swimming like crazy, and some of them even jump out of the tank. So cortisol is driving this behavior. So here we have a treatment that's inherited transgenerationally that we can rescue with cortisol. What about generation 4? I'm going to skip one slide just in the sake of time. We saw similar decreases even in generation 4 in the cortisol, but it wasn't that exciting. And the experiment we did is to again treat them with a second drug, venlafaxine, that some of you will know. It acts on norepinephrine and serotonergic neurons. And just for the sake of time, I'm just going to focus here on the males. So here's the same groups, generation 4. Give them venlafaxine for 6 days, grow up the animals, look at their stress response, and breed them again. So what happens? You have a sensitization to venlafaxine. I'll show you right here. Here's the stress response, lower in the fluoxetine groups and intensified when you give venlafaxine. It's okay. It's not that impressive. But breed these animals and see what happens, and then you see something I think is quite profound. It sensitizes the animals to inhibition of reproduction. Here's the spawning rate. So you put male and female together and you ask them to lay eggs. And they are pretty normal. I told you that at the beginning, even on generation 4, except if you treat them with venlafaxine. 3 out of 13 couples gave eggs. That means 10 out of 13 did not breed. And we know how to do this, and we're very convinced. Those 3 couples that bred are normal, but the 10 couples did never breed. So ancestral fluoxetine is having an effect on the next treatment generations later. So how are we going to deal with this in our pharmacology? Okay. So I hope I've convinced you that cortisol disrupts, antidepressants disrupt cortisol. It's important for us, and it's important for aquatic animals because fish, just like humans, need cortisol for the adaptive response, the classic Hanselier adaptive response. But remember, the problem that I've shown you is hypocortisolism. We now know that too little cortisol is associated with burnout, chronic fatigue, fibromyalgia, immune disorders, and post-traumatic stress disorder. So chronic high cortisol is not good for us, and chronic low cortisol is not good either. So we're faced with a problem right now. What are the implications for humans? Well, here's these studies from British Columbia again, UBC. They've been studying. These are older studies, but they're continuing even now in 2022. Internalized behaviors, anxious behaviors are increased in children coming from mothers who are taking SSRIs during pregnancy. It's not a direct link, but it raises concern. Gross motor function in a 10-month-old child is decreased in moms taking SSRIs. So we have some recapitulation in the fish model of some of the behaviors that the UBC group is showing in children. Okay. And here is the second last slide, and thank you for your attention. I've put a little history here. The very first data slide I showed you was in 2013. All of the different generations took us till 2016. When I showed you generation 4 having a sensitization to venlafaxine. Look at this. Here's the generations of humans. 1986 is when fluoxetine was put on the market. We're somewhere in the middle here between F1 and F2. I've shown you that fluoxetine's effect persists for generations. We do not know if the fish is a predictor of the human, but here are the dates that we can look forward to in the future. Perhaps we have to be much more careful with how we use these chemicals. That's my point. So final message, chemical exposures of all sorts, therapeutic, non-therapeutic, pollutants, what have you, may not only have repercussions and effects in our own lives, but also in our descendants. Thank you very much.
Thank you, Vance, for this nice presentation. Just a moment. Oh yeah, I'm here. Thank you for sharing your data, your beautiful and scary data. It's the second time that I listen to you about this research in your lab. It's really caused me some feeling that really I don't know how to manage that. So thank you very much. And we already have some questions. Paulo.
I see that.
Can you see also the chat?
Yeah, I can see Paulo's question.
Yeah, Francois. So the first one is Paulo. What about metabolism? For example, are there changes in weight gain, grow, etc.? So other kind of changes in these
Yes,
animals?
good question. So in our original studies, we looked at food consumption and growth rates in adult fish, in goldfish, and acute exposure decreased food consumption and weight gain over a month. kind of like some people report when they're first taking fluoxetine. In this particular developmental study, we saw no difference in body weight ever. There was no difference in weight gain. The animals were quite similar. So no change in metabolism that we can see in terms of growth rates or things like that. I hope that answers your question.
So Vance, in the same line of Paulo's question, I was thinking, since you find so many differences in cortisol levels in generation F2, F3, and also in the genes related with steroidogenesis. So did you look at other kind of steroids, so sexual steroids? So should they also be changed?
No, they're not. These animals grow very well. They reproduce very well, except after the venlafaxine. But that was our big surprise, because we had thought we would see the same thing as we saw in adults, but no reproductive effects. And when you measure whole body testosterone and estradiol and another steroid that's called 11-ketotestosterone, which is a main androgen replacing DHT that you would find in humans, no difference at all. So these animals seem to be fairly good at reproducing, except in the F4 that I showed you, and no changes. This seems to be very specific for the at least steroid levels on the cortisol.
Okay, thank you. So that's a good news. There's more questions and comments. Professor Francois only made the comments that was brilliant, your lecture, very didactic, and anxious generating message. Exactly. So we feel anxious in listening to see those data. Professor Vanessa? That's okay. Yeah. Professor Vanessa also made a question. Thank you, Dr. Vance. Nice presentation and results. Have your lab already investigated epigenetic mechanisms underlying fluoxetine, cortisol, and behavior disruption?
Great question. The answer is a little bit. So it looks like an epigenetic inheritance. And we haven't delved into anything like DNA methylation or histone modification yet. However, we observed some years ago that you can alter the expression of a certain number of microRNAs. MicroRNAs, of course, being another epigenetic mechanism. So what we found in the zebrafish as well is that you can alter some of the microRNAs that may be linked mostly towards metabolism and other things like that. But it looks like we can alter the microRNAs. Now, if you do a crossbreeding, fluoxetine male and normal female and fluoxetine female with normal male, we know that this is largely driven through the female. So different from mouse, different from Tracy Bale's data showing that the epigenetic inheritance of stress is through the sperm. This seems to be through the female. Our data is not very strong, but it's still nevertheless clear. And it looks like the mother is able to deposit different levels of the microRNA in the egg. So we think that's setting it up. We don't have very strong data, like I said, but the microRNAs in the egg of a fluoxetine mother and a control mother are different. So we think that might be one of the initiation steps for the epigenetic inheritance. We're debating right now if we want to go down the route of epigenetics. So the one thing that we have done is we looked at the brain and doing some RNA sequencing on the first generation. And we can see 6 months after exposure, some of the typical epigenetic machinery is altered in terms of messenger RNA levels. And that's the work of Amin Nozari, who just graduated, and we're just in the final stages of publishing his second paper that will describe this. His first paper showed that other chemicals can also alter the stress response and some of the epigenetic genes and some of the genes implicated in neurogenesis and what have you. So it looks like there's going to be some effects through the epigenetic machinery as well. But we have a long way to go to really answer your question properly. It's in our mind, for sure.
Okay. Thank you, Vance, for this answer. So the next question from Adriano, he makes a relation with chronic stress. Thank you for your amazing lecture. How previous exposure to fluoxetine could influence later in life response to a severe stress exposure, such as chronic unpredictable stress. It would make subjects more resilient to develop stress-related response, such as depression-like behaviors?
Fantastic question. Don't know. I do not know. However, how could we do this? I don't think the stresses we've used are predictable in one sense, because generation later we just grab the fish and throw it in the tank, or we just grab it with the net. It's not a predictable response or stress in the sense. So I think it's not going to be adaptive, but I understand your question. It would be very interesting to do that type of experiment. These are very difficult experiments to do, as you can imagine, trying to maintain the animals for so many generations without anything going wrong, and almost everything affects the stress axis, as you know. But this is a very, very thought-provoking question, something to think about for the future.
Good. And Paulo and Christina made comment that they are impressive with the information you told us, that there is no this current generation cortisol level is not affecting the other levels of steroids.
Well, I mean, it's very clear that it's through the stress axis. I don't think it has to affect the other steroids. Again, let's contrast these data with what we know about adult fish. When we do, because we were surprised as well. When you give an acute treatment over 7 days to an adult fish, you can change the steroid production levels in response, a male in response to the female presence, different scenarios. So that's what we got very excited, and we thought we would see a transgenerational effect on reproduction. But this early exposure does not seem to be altering any of the processes related to sexual development. They are just fine. So there's something different from the adult acute exposure to the embryonic exposure. Very intriguing, but we don't really have a good idea on that.
So in this sense, then, about the difference you find between female and male fish. So how was the explanation for that if probably the other sexual steroids are not involved?
Well, there's more to life than sex steroids, of course, and the difference between males and females. I don't have a specific answer for this, but it's not uncommon for animals and rats and humans that females deal with stress differently than males. So often we think of it as a different in the estradiol levels, where there's an altered response to the sex steroid, to the stress because of higher estrogen levels. And in fish, this species, the females do have higher estrogen levels, but not by 100-fold or 10-fold like in humans, just only 2-fold. So there could be some of that. There could also be some differential development of the circuits that are different between males and females that could account for that. But yeah, it's quite intriguing, but it's very clear in the males and less clear in the females. However, this is the interesting part. When we do the back crosses, this effect is inherited through the mother. So the males are affected more, the sons are affected more, but it's the mother that's transmitting the information through the microRNAs and probably other epigenetic mechanisms. So for example, if I take a female that was treated as an embryo, PROZAC, and compare it to a normal female, cross with either a PROZAC male or a normal male, we can figure out that it's the PROZAC mother that transfers the information. It's not the PROZAC father. So the offspring of that mother has a reduced cortisol response. It's incredible.
Yes.
And that was published by the same student, Marilyn Viracheng.
Very nice job.
Oh, fantastic! Yeah.
Let me see if you have more questions. Not in the chat. There is a lot of congratulations to you for your presentation, excellent presentation. Well, there's one more question now from Victor. We saw from your talk that the CIP system, CIP system, altered by fluoxetine can cause problems with cortisol production. If so, can other drugs that similarly alter the system cause the same problem? Do you have evidence of other medications?
Super question. Can you move to the next slide? After. One more. Okay. So here's where we are now. This is Amin Nozari's work. And we asked the question, will other chemicals inhibit the stress response? And this is a little bit of a cherry picking because we had evidence from other animals that this fungicide, vinclozolin, this is the famous one from Skinner's group, bisphenol A as a pollutant and xenoestrogen, and dexamethasone as a synthetic steroid. These are just exposed overnight. And here's the same setup. Stress response in embryo, larvae, inhibited by bisphenol A, inhibited by vinclozolin, inhibited as we'd expect by dexamethasone. And with this experiment, we're trying to develop a method to predict the future. And we're using a GFP model. Here's the GFP levels of the stress response, and it exactly mirrors the cortisol response. So we don't know the mechanism, Victor, but we're trying to figure out which chemicals we can screen where we can delve into the mechanism and ask the question, if you were exposed to bisphenol A as an embryo, would your stress response be altered over the generations? So that's where we're kind of doing that right now, trying to develop a predictive model to understand, does a short-term drug effect persist over generations? Okay. Now, specifically about the CYP system, we do not have that answer yet, whether these other chemicals would have similar effects. The first step is to try to develop a screening method, and that's what we're doing with this. And a new student, Emmanuel Monez, is just starting her PhD on this to pick up from Amin's work to try to hone in on a new test to be able to predict the future.
Good. Very nice. Vance, and we think about the all this data show that we have a lot of environment problems. Of course, you show the data in fish, but probably other animals, aquatic animals, are endangered. And also the water that we have at our home, that we arrive at our kitchen, for example. So we have a lot of fluoxetine and other chemicals, but basically in your data, so we are also exposed to high levels of fluoxetine, probably by the water that is in your home.
It doesn't seem very high.
Oh no. So
No.
Of course, I guess that depends of the different countries and the places.
It does get removed. It does degrade eventually, and the filtration systems aren't designed to remove it, but some does remove. And there's been several studies of tap water, and you can detect fluoxetine and many other chemicals in the water. It really depends on the source of the water, but we all know where it comes from because we're putting so much sewage into the rivers, and we take the river back to make the tap water. So there is very low levels being detected. Very low levels being detected. I would say, without knowing too much, that it's not going to be that dangerous. But when I gave some years ago, when I gave a different lecture to the public, and I asked the young mothers, what level of fluoxetine do you tolerate in the drinking water of your child? You know the answer. 0. And they don't have to know that it's harmful or not harmful. Having drugs in your water is wrong. Every person in the audience, in the public, says that. So it's a different question, but it's going to be there in some places. And I don't know if fluoxetine is the one to be worried about. I think there's many other more serious ones. But we are getting some of these things showing up in city tap water now, even in places that you think are clean, like the middle of the Amazonas, where very few people, or in northern Canada, or wherever. Anytime you have people taking medications, you're going to find it in the water. Now, that doesn't mean it's end up in the tap water, but it might.
Yeah.
Very low level. Very low.
Because I guess maybe in your country, in Canada, the system of the treatment of the water should be special. I don't know, but here in Brazil, actually, the city I live in, Curitiba, it's quite poor. There is a system that can remove some bacteria, but there's no filter to remove this kind of chemical compounds.
Only in Switzerland and Germany do they have that. It's extremely expensive.
I can imagine.
Billions of dollars it will cost to do the reverse osmosis.
Okay.
So what people are doing, and in Canada, is trying to find the type of sewage treatment that's currently working that removes most of these, because there is no depurification plant in Canada designed to remove pharmaceuticals. No way. We are really slow to change in North America because it's so expensive. But there are certain systems where the secondary and the third treatment degrades many things. So that's a good news. So with the treatments and the digestion, a lot of these chemicals do break down. So that's good news. So what's happening in Canada is they're trying to identify those sewage treatment plants that are the best at removing these things, and then the other ones can change some of the processes without spending billions to build new ones. But it's a big challenge for sure. It's a big challenge for sure. I think another thing to consider is in new cities and new developments, do we have to be using very good quality water in our toilets and then putting that good quality water in the sewage and making it dirty and then trying to clean it again? Why can't we use dirty water in the toilets and have a special treatment plant for that type of water and a different treatment plant for the clean water from your bath, for example? The bath is not a big problem. So these are other questions that engineers are trying to address all over the world, also in Canada, also in Brazil. You have some good colleagues in Brazil working on pharmaceuticals in the environment. So for those of you that are interested, you can contact them and perhaps make a collaboration, because these are really important questions. The impact of the pollution on the human.
Exactly. So, and how about the regulations authority? So, do you know if your research is impacting some regulatory laws in Canada or influence the authorities?
I think some of our data contributes to the general knowledge of what levels affect the aquatic organisms. But this particular study that I just described to you on the transgenerational, and this test that I'm showing you on this slide, is becoming very interesting to the authorities. And the first step we're doing right now is we've developed a really nice collaboration with scientists at the Health Canada, which is the national health agency. They have health researchers. And we're trying to develop this test that I'm showing you on this slide to be able to screen many, many chemicals of interest. Because we have a chemical registry in Canada, and there's a new substances list or old substance list, and some of these chemicals have never been tested for endocrine disruption. So we're now setting this up, the new student, Emmanuel, that I mentioned, with some colleagues at Health Canada. Some of the collaborations were with a woman by the name of Carol York, who's now with us at the university, and Dr. Errol Thompson at Health Canada. We're really putting our heads together. Can we use this type of test to screen those types of chemicals? Because the stress axis is very conserved between fish and frogs and cats and dogs and humans. So
One health.
Yeah, one health. Exactly.
We have two more questions from the audience. Vance, from Professor Maria Cristina. Have you explored changes in GR expression in your model, glucocorticoid receptors?
I'd have to look at the gene list. I do not know. I don't remember. There's many hundreds of genes changed. I do not remember specifically. We have not specifically looked at a GR distribution by immuno or in situ hybridization or anything like that. Very good question. Certainly something we can think about, especially when we're talking about the behavior change. Perhaps it would be very interesting to look at the GR expression in certain brain regions. Thank you for the question.
And we have one more question from Professor Roseli. Thanks for your presentation. Consider the importance of the cyclic AMPC signaling cascade in serotonin action, cortisol production, and stress response. Fluoxetine treatment could promote change in AMPC signaling in the next generations?
Good one. I don't know, but I would guess maybe, because of the reduced ACTH response. It's in vivo, so it's a little bit hard to predict, but one would think maybe there's an uncoupling or a reduction in the CREB or cyclic AMP response binding elements or something like that. a reduced sensitivity to ACTH, perhaps. Yeah, another great question, but we have not delved at that level at this point. Maybe with this larval model here, we can get into the mechanism, perhaps. I'm not sure. I'm not sure.
Okay. Thank you, Dr. Vance. So I guess that was the last question here from the audience. Maybe we have more questions, or somebody would like to make a question by a voice. Otherwise, I just thank you very much, Vance, for this very nice presentation, for showing us your data that's quite impressive to us. And thank you again for your good job and for being here with us in this morning. So
Okay. Yeah.
very early for you.
Yeah.
And now I pass the word to Professor Paulo. Thank you.
Thank you very much.
Thank you, Alexandra, and thank you very much, Professor Vance, for the nice lecture, bringing us with new and important information about the use of antidepressants, in this case, the fluoxetine. And now I will finish this session taking em português, ok? Alexandra, você pode colocar nos slides finais aí para a gente?
Já coloquei, Paula.
Tá, ok. Sim, sim. Então, Alexandra, agradeço a sua colaboração, né, em moderar esta sessão e também em ter indicado o professor Vance, né, que trouxe um tema bastante interessante, né, mostrando essa questão da poluição ambiental e a influência que traz para as gerações, né, dos nossos medicamentos, no caso, os antidepressivos. Em nome da coordenação do SIG Farmacologia Terapêutica, então, agradecer a sua importante colaboração, que sempre tem participado fielmente às nossas sessões do SIG Farmacologia Terapêutica. Agradeço também a participação de todos vocês que estiveram assistindo esta sessão, lembrando a todos que esta sessão é gravada e que será disponibilizada no site da SBEFT, né, no link que aparece aqui no slide, www.sbeft.org.br/sigfarmaco. Seguinte, o próximo encontro, a próxima sessão será no dia 20 de abril, né, onde teremos a palestra do professor Leandro Agatti, da Science Valley, sobre moderação da professora Cristina e do professor François, como coordenadores e moderadores da próxima sessão, onde todos estão convidados novamente a participarem, né, também com um assunto bastante interessante, bastante inovador, que é sobre o modelo de negócios na pesquisa clínica global. Lembrando a todos novamente da importância de registrarem suas presenças nesta sessão, acessando o link www.rut.rnp.br/presença, selecionando o SIG Farmacologia Terapêutica e a senha desta sessão, que é 55973. Agradeço mais uma vez, então, a participação de todos e nos encontramos na próxima sessão. Tenham todos um bom dia e uma boa semana.
Bom dia, obrigada, bom dia a todos. Para parar a gravação, Paulo, automaticamente aqui.